All right, welcome everyone to Symbiota Support Group, September 2023 edition. We're excited to have you all here. Just a couple of announcements to start us out, or kind of really one announcement right now, is that BioDigicon, the Biodiversity Digitization Conference put on by iDigBio, is happening next week. It's a completely virtual agenda, and it is completely free to join. So I would highly recommend taking a look at that at their wiki, looking at all of the talks they have lined up, and being sure to register and participate in that meeting. So that's going to be again happening next week. We have a couple of Symbiota talks that are going to be happening there, and it's going to be a good time.
Awesome. Thanks, Katie. And I will add to that, I think I saw the schedule this morning, and Jorrit, who I'll introduce in a little bit, looks like you're giving a talk next week kind of on the same topic. So if you can't get enough today, tune in again next week, and there'll be more on this at BioDigicon next week. So something to keep in mind. All right. So any other announcements before we get going from the audience? I don't think I see anything in the chat, so we're good. Okay. So thanks for joining, everybody. Today's topic is linking to genetic data in your Symbiota portals. And so today's Symbiota support group will be a little bit different than some of the other ones we've done before, in that it's going to focus on a case study. And so we're first going to introduce the case study. What is the case study? What was desired from this case study? What resulted? We'll talk about how do we accomplish this. So we're going to kind of give you the bigger picture, and then kind of pull back and talk more about the steps we took to get from A to Z. That'll be sort of the heart of what we're doing today. And then we'll focus on some other methods for getting genetic data into your Symbiota portals. Then we'll briefly touch on the extended specimen, and then we'll wrap up with briefly talking about some next steps and opportunities. And then, of course, as usual, there will be time for a discussion, question and answer related to the presentation, or any other questions you have about Symbiota. The hub will be there to help answer those questions. So, intro to the case study. So to give you a little bit of background about what we're going to be talking about, so something that happened, say in June, about 3 months ago, some of us were at the Digital Data Conference hosted by iDigBio at Arizona State University in June. And Jen Yost, the curator of the Hoover Herbarium, who's also our community lead, brought up in one of our sessions on the extended specimen that she was having this recurring issue. So yeah, backtrack 3 months, and that's kind of where we're setting the stage for what we're going to be talking about today. So Jen brought up specifically that she has issues with finding and round-tripping genetic data back into Symbiota. So often people come to her collection, or she'll send them alone, and then they'll do an analysis, genetic data will result, but then she doesn't necessarily get that data. And so Jen was asking, how can I get this data back into my Symbiota portal? And so this is sort of the origin story for what we're going to be talking about today. And so the data that were involved. So Jen is curator of the herbarium, so it's mostly herbarium vouchers specifically associated with the OBI collection in the CCH2 Symbiota portal. And if you go in and look at that collection, there are 94,000 specimen records, so quite a few records with potential to be linked with genetic references. And then the other part of this is that there are OBI records in GenBank, which, if you're not familiar, is the NIH genetic sequence database. And I believe we have a rep from there in the room today. Just fun fact. And so what this would look like if Jen were to go and try to look for her specimens in the GenBank database, she might type in OBI or OBI voucher. And kind of what we're just trying to show here is there'd be a lot of results. And so it's overwhelming for Jen to figure out okay, now what? How do I get this information from this portal into my Symbiota portal? And that's kind of in summary, the question that she had. And so her results is that what did she want? She wanted specimen records in CCH2 to be associated with her GenBank data in the GenBank database. And so if this was accomplished successfully, this is what it would look like. She would go to her public record in Symbiota, and there a little genetic tab would show up, and the information would show up on the record. So I'll just show you what this looks like. So here's this record in Symbiota. If you successfully link genetic data to your specimens, this tab will appear, and you go to this record, and there might be one or many records on this one individual record. So that's what she wanted. And then, of course, if she's already publishing to GBIF, she also wants this information to go to GBIF, so that is associated with the specimens in that database. And so for the same record, this is what it looks like in GBIF. So if this is successful, she should be able to go to this record in GBIF, scroll down, and then that same information that was in the portal should be under occurrence under associated sequences. So this is kind of like the end result that she was hoping for. And so how do we get from, A, no links, to B, many links in your portal? And so we've invited a guest speaker today, Jorrit Poelen, to talk about the GenBank to Symbiota round-tripping workflow that he came up with. I just want to introduce Jorrit for people who don't know him. So Jorrit Poelen is an independent software and data engineer. He's a Ronin research scholar and research affiliate with the UC Santa Barbara Cheadle Center for Biodiversity and Ecological Restoration. Jorrit maintains an active interest in open science and biodiversity informatics. He also contributes to the Global Biotic Interactions Project, also known as GLOBI, the Terrestrial Parasite Tracker Project, Big B, and the Open Trades Network. And over the course of the summer, he's also worked with Jen and Katie on this particular project that we're talking about today. So Jorrit, thank you so much for joining us. And just want to point out that Jorrit is really good about documenting things in GitHub. So I put the link to the little GitHub repository he's put together for this. I'm going to stop sharing my screen, and Jorrit, feel free to take it away.
Mm-hmm. I think screen sharing is disabled.
Oh, let's see. Who is the host?
Oh, yeah, I got it.
Awesome.
Let's see, I can do this. Yeah, thank you so much for having me. And it's been really a lot of fun working with Jen and Lindsay and Katie to work through this super fun use case. It really tested a lot of my skills, mainly my communication skills, which is a work in progress. And I also got to try a couple of different ways to try and get to the result where we ended up, which is what Lindsay just shared, like ways to link genetic records that are available in GenBank from Symbiota, and I'll share some notes on that right now, and please feel free to interrupt you if I make no sense whatsoever, or if you have any questions. I do realize that I come from a technical point of view, and I also work a lot at my kitchen table. So sometimes I need some help to explain what it is I'm really trying to say. And Lindsay, Kate, and Jen have been very patient with me as I'm sort of working through this. So I'm very grateful for that. So this is the moment. This is actually the screenshot of the moment where Jen expressed her desire to link her specimen to genetic data. And so she was saying this here, and there's actually a recording of this, which you can find in the README that's shared on the GitHub page. So I was on the panel there, and I think this guy is also in the call, right? Is that that's and
Conrad is here.
Our guest, or my fellow guest, Conrad. So I'm hoping that this is going to spawn some discussions that help me to better understand how to improve what I've done or what we've accomplished so far. So yeah, so this really came about at this workshop. It took a ton of work to get here in the first place, to be able to have this conversation in the first place. Loads of organizing, loads of networking. So there's a lot of time and effort that sort of went into this just to get to this place. And all the people in the audience helped make that happen. So I want to emphasize that, because I think the skills needed to do these complex data integration issues are not just, or are partly technical, but mostly social, and rely on an active community that's willing to try out things and willing to put energy and time into making seemingly impossible things possible. I'm sure you all have your individual stories around this, because you're all working with digital data in biodiversity in some way, shape, or form. So I was really glad to see that this thing was recorded, and if you ever get a chance to watch that video, I would really recommend it, because Jen really knows how to articulate her desire quite clearly, in my mind. So I try to document what I've done in collaboration with Lindsay, Caitlin, and Jen, and in the form of sort of like an article. So I'll basically scroll through it, because I sort of felt that creating a PowerPoint would be like sort of like a thorough way, and I rather have something I can reuse. So all this is available on GitHub, and I can share some links later. So here's a little bit of an introduction. As we know that there's a lot of specimen records available, and they're now also a lot of them have information stored through GenBank, and those links aren't always obvious. Some systems are very nice to sort of do the round tripping almost like out of the box. So Arctos is a good example of that. And for other collections, it's a little bit more, the links are there, but you really have to know where to look. So that's what I've learned, is that if I work with the people that work in the collection that is being linked, a lot of time and effort can be saved, because they know exactly how the data is used and how, like, what things mean. So that was really quite helpful, and a very useful investment of time, just having that half an hour or like that hour of back and forth through email or through Zoom to better understand the workflows. So that was really important to me. Otherwise, I could have not done this. This would not have happened. So not understanding the workflow is just, or not understanding or knowing the people behind the collections would make me very hesitant to sort of do something like this. So one of the first clues that sort of came up was the institution code for the collection that we were working with. So we had a very specific use case, right? Like one herbarium wanted to share or link their collections, and OBI came up a lot. So that's the sort of the airline code for the collection. That's how I see it, or the airport code, and that's something I wanted to point out really quickly. So here's a bit of the challenge here. So you have GenBank, which is giant, right? Openly accessible digital resource, and we have this OB collection that also has a digital catalog, and we're trying to figure out what this overlap is here, right? Like how they're connected. We know that there's some connection there, but we not like, so intuitively this makes sense, but we're trying to figure out like how this works, right? Like what does this look like? So this is one of these one of the examples of like what does this look like. In the real world, they're really separate entities with their own teams and their own, support staff and technology and all that jazz. But in our minds, it really looks like this, right? Because it's all like it overlaps in knowledge, and there's links and all that stuff. So how do we get there, right? So one of the first things I did was to try and ask for an example, or look for just one example that sort of shows something about what's in the collection. So here was one record and their HTML landing page, right? So this is what you see in the browser. I think if the internet gods are with me, I can even pull this up. I don't know where it went, but yeah, it's here, right? So I'm lucky that the site's still up. I don't really know whether anything changes because it's really hard to see what the difference is between the screenshot and this thing. I can do some visual inspection, right? But this already shows the first step to integration. Like what kind of source data are you gonna use to integrate from, right? So HTML is not the best in my mind. Some people really like it, but you know, but at least we have an example here, right? So we have an example here, and then you can look at the you can go to the linked record or some of the linked records on GenBank, and then you also get HTML. So I guess that's a common common theme that they have HTML landing pages, because it's, you know, HTML is a very popular way to share stuff, right? So it's here. Again, I can't really see whether the screenshot I made earlier is any different from this one, right? Because, well, I can do sort of character by or pixel by pixel comparison, but it's kind of hard to see the difference. But it sort of looks, you know, somewhat similar, right? And again, I was lucky enough that GenBank is still up and running and all the infrastructure is there. Most likely, you know, according to the evidence we've gathered over the last 20 years, this site is going to be either moving or changing or something like that in the next 3 months, 5 years, 10 years. Conrad probably knows more about the strategy, but that's like a normal, natural process, right? So just like the Symbiota portal is being upgraded all the time and they sort of move around. So that's why I actually took a screenshot, because you know, that's one way to version the data, right? Just like very brute force, take a screenshot, take a picture. It's almost like iNaturalist for the internet, right? So you take a screenshot of your data. So this may or may not look familiar to you, but to me, I'm already seeing some clues here of how things may be linked. But if you look at the textbook examples of like how things should be linked, you would expect some kind of link here that you can just click on and then go back. Sometimes that's the case, but in this case that's not there. But the person putting this in took a lot of effort and time to carefully record what they thought was identifiable information for the linked specimen, right? So rather than dismissing this as saying, this doesn't link, I was kind of excited to see this in there, right? So I had my example. So now I was looking at the things that might be of interest. And I don't know if I can zoom in. Yeah. So here's some of the, I've sort of highlighted some of the things that may be useful for linking stuff, right? So some taxonomic information seems to overlap with the specimen record, right? And then the collector was there, and then some kind of number is being used here. I sort of later learned that that's a collector number, which is also referred to as a record number, which is very confusing, or was confusing to me, and now I know that they're sort of used in that way. And then also some secondary collector number here, or catalog number over here. I didn't highlight it, but you can see that there's some traces of that here, and they sort of smush together the institution code and the collection code. So, and also the collection date, right, is there. So there's all these hints there that we might be able to use to find more of these that may connect to these. That was really the challenge, right? To take these millions of records in GenBank and try to filter them down to candidates that might link back to the OB records. So now we have that example. So I had some hints, and this is also after some conversations, right? So first I knew that the internet of today is not going to be the one that will be there tomorrow. So, you know, starting from this idea, right, where you have GenBank and OB, and you want to, you know, see what the overlap is, you first want to sample. You want to sample it. You want to version the GenBank version, right? And I don't want to just version one thing. I want to version everything, right? Because I don't know which record in GenBank is going to be the one that's a candidate. I can try to poke around and sort of click around and sort of get screenshots and make screenshots of these things, and then go over that. But that would probably, I would probably die before I get like halfway down all the records. So I had to figure out a way to version GenBank, like all of the, you know, likely candidates, right? And then ideally filter down to this sort of bit that might be useful, and then we can sort of put it together, right? So it's like version, filter, so you reduce the number of candidates from millions to hopefully, you know, fewer, right? And then you do the same with OB stuff, right? So you get all of the OB records and then version them, because OB, just like GenBank, is something that sort of changes and gets updated. So you want to start with a version copy and then filter it down to select the ones that might have GenBank records in it. And then you sort of merge it, and then you get this. So we still don't really know what this is, right? Like, how do you write this down? Like, what does this look like? So I'll get to that later. So acquire and version, right? So first you have to get GenBank, and then you have to like take a snapshot of it, right? So first you have to catch it, and then you have to put it in a jar. So my tool to do that is a tool called Preston. It's just like a sort of a save as, but then a little bit more complicated. And what I made it do is I made it go to the GenBank archives, which is a very cool way that they have. It looks like something from the, not even from the 90s, but something from the 80s. And if you look at this, it sort of looks like, yeah, looks like, what the, you know, what am I going to do with this? But it was really nicely documented, so you can actually read the README, and they have a very neat, very detailed description of what's in there. So with this information, I was able to create, actually, I was already able to pre-filter GenBank to only include the part of GenBank that talks about plants, because I knew that the OB was an herbarium, so I figured there were plants in there, so I could sort of ignore all the other stuff. So that's the first assumption, right? So I was able to reduce, you know, maybe like, yep, I was able to filter down already quite a bit. And then I basically grabbed all these files. They sort of look like this, and they're just text files. So if you look at them, they're just text files with, and I'll show in it. And these files basically look like this, right? It's just a text file that looks like this. So it's, and then sort of copied into big sort of bulk things. So there's no magic there. So if you have a notepad, then you can basically uncompress these things, and then you'll see all the records in there, right? So with this, I was able to basically download over 200 gigabytes of data. I was able to version it, package it, so I can cite it, put it on my hard disk, and also have a copy at ASU, which they offered to keep. So I have a secure version copy of this thing that I'm working with. So there's no rugs being pulled from underneath me. I'm trying to reduce variability, right? So here's some examples of how I did it. And without getting too much into details, this is basically a unique identifier that basically identifies the entire thing. So it's the entire corpus, the volume that I versioned, that includes all these records and also describes where it came from. And this is also a key I can use to basically make an identical copy and move it around. So it's a very handy way to make copies of things without having to worry about their current location. So I did something similar for the OB herbarium records. So they publish their Darwin-Core archive through Symbiota, and that was also versioned and tracked. And so I packaged that very similar to what I did here. So now we're sort of over here, right? So we have a version of OB, we have a version of GenBank, and looking at the time that it took me to explain this, this is actually a big step, right? And it's easy not to do it. Most people don't do it, I think, because yeah, it's very convenient to assume that the internet will stay exactly as it is today, right? So I can use a web API or some kind of HTML page and use that to do my linking. But if I do that, I don't have the foundation I need to do rigorous linking, I think. Okay, so now we're going to look into filtering this. So we have a copy of this. I made a tool that allows you to stream all the text into one stream of data, and then you can filter it by records that only contain the keyword OB, right? That's it. That was my brilliant filter term. And so Lindsay showed what happened if you use the OB term into the search, right? The web search. So then you get like something like 200,000 hits or something, 20,000 hits. So that's a lot of hits to basically scroll through and click through. And when I did this on a more basically machine readable version of this in my own custom way, I was able to reduce the number of links down to hundreds, right? So all of a sudden I was able to go from millions to hundreds using a sort of an understanding of the format and the data, and also having an understanding of what kind of keywords might be useful to hone in on the candidates. And then it's very tempting to get more advanced and more fancy and do AI and machine learning and all that stuff. But if it's just 200, you can just look at them and document the candidates and share them with the collection staff, which I did. So Katie was kind enough to have a look at a data sheet that I created. You can see this is actually the sheet we use to review the candidates. So I shared this data, which basically contains the reference to the GenBank record, the definition, the content of some specific records that are, or some specific fields, right? So the specimen voucher is that thing that we saw earlier in the GenBank record, and so you can see that they're very different, right? So you can see, but there's patterns there. So this looks like a name, it's like a number, and then institution code is there. Sometimes the OBI thing doesn't show up in the voucher, but it shows up in the definition. And this thing derives from, this is actually the exact location where it came from. So this is the thing you get when you do versioning. You can point exactly where it came from. And if all things work well, yeah. So this is the exact copy of that record that was picked up. And if you look at OB, there should be some kind of OB in here. So once I showed this to Katie was like, this is not one of our records, right? So she was basically sifting through it manually. She was doing the what they call the clerical review of the candidates. And there were no's, but also yeses, right? And with this, we were able to go through, and for each of the yeses, I was able to very quickly identify which patterns you could use to link to the exact specimen record. And I also included the query needed to get that unique thing in there. So there was a little bit of back and forth, right? I know I'm sort of sharing a lot of details here, but I'm trying to get the gist across of the method. And the gist is you basically try to reduce, you capture the data, you version it, you try to filter it down as much as you can to the candidates, then you have a conversation, and then after a couple of back and forths, hopefully you get to a list of links that you can incorporate. So now we have this list of links that we can incorporate, and here you can see that we've added the landing pages for the associated specimen records. And with this list, Katie was able to do some tricks and do a batch import, I think. Is that what you did, Katie?
Yeah, pretty much.
Mm-hmm. And then you were able to pull this information in, if I understand it correctly, and populate it on the Symbiota side. So now, if you look at the genetics tab, you have all these identifiers there. So the link from Symbiota to NCBI is there. Obviously, it would be nice to go back also, but we've at least made one step, right? And now you can tackle another collection, or you can tackle another link method, right? Or direction. And looking at the results, you can see. So I versioned the OB collection before and after the linking. So I was able to really precisely see what changed, right? Because I'm able to say, hey, show me what it looked like. Show me what the records look like before, and show me what it looked like after. And because they are so detailed and versioned, I can use all sorts of tools that are readily available to pick up on these diffs. So here's just a visual representation of that here. So this is before our experiment, and here you can see the associated sequences are empty for this particular record. So this is coming straight from the Darwin Core archive that was published probably in June somewhere, June or May. And this one is a more recent copy where you can see the labor of love, right, that we put in here. And you can see the associated sequences pop up in the data, in the core, like the raw data. And then, as Lindsay showed, this data is now propagating to things like GBIF and probably also to iDigBio portals. And hopefully, it's going to make it, it's going to have some kind of benefit to folks that want to use this data and find it through the links. So I think I just want to stop there because I'm also very curious about the discussion. One of the things I do want to mention is that I wrote sort of like a I wouldn't call it a complaint letter, but sort of like an open letter to, it was addressed to Lindsay and Katie and Jen, which was about the finances of it all. Like, who's going to pay for this, right? And how does it work? And I made some kind of silly calculation of, so here's the letter, right, where I sort of break down my the time we sort of spent on it, and then do some calculations of how many, what the reward was. So actually, at another workshop at Gilbert's, gave me a tub of cookies. So that was sort of the challenge. Actually, also in the workshop, I said, hey, I'd be happy to do it, but I'd like to have some coffee and cookies. So I got the cookies during at another workshop, and I counted them, and there were 78 cookies. And I was able to express now what a link costs, right? What how many cookies per link you'd have to spend to make this work. And so obviously this is like a very silly example, right? But I calculated like 3 cookies per association, because it was, we found 25 GenBank links. May not sound like a lot, but it's probably a lot of work that sort of went into that to create these records in the first place. And if you extrapolate that and basically say, hey, we have 0.03% of the records having GenBank associations, and then you extrapolate it, then you can see that you need about 2,300 tubs of Trader Joe's ginger cookies to make all the links. So, this sort of opens up the economical question of this, like, what is it worth? What are people willing to pay? Who are they going to work with? And, who's going to do it, right? And I want to end with this quote that I sort of learned as a kid, visiting the neighbors. They had this quote on the wall saying, everybody wants to go back to nature, but no one wants to walk, right? So I sort of translated it into the digital extended specimen world, which I sort of translated as, everybody wants to have a digital extended specimen, but somebody else has to build it, right? So with that, thank you for your time. And I'm very curious to hear what I can learn from your comments and questions. Thank you.
we can pause for questions here, or I can kind of wrap up the slides, and we come back to some more time for questions at the end too.
Yeah, please. I'm trying to stop share, but for some reason it's not working. So maybe you can force me out.
I do not see your screen anymore, so it doesn't seem like
excellent.
good. Conrad, I see you've unmuted. Was there something you wanted to say right away?
Yeah, I've got a question. So that GenBank accessions, was that done by a third party or by yourself? Who made the submissions?
These were done by third parties. These were done by researchers that have used these specimens over the years. And the example I used was a specimen collected in 1966, I think, and it was accessioned in 2011, or like if I remember correctly. I'd have to look at the record again. But then I don't know who that person was. Maybe Jen or Katie or
yeah, I guess that's common. You know, that's a reasonable assumption that it was somebody else. Because I'm thinking about, you know, possibly updating those records so that the format for the specimen voucher is corrected, and you know, if it's Darwin core format, then you can link directly from the GenBank record out to the herbarium.
Yeah.
Not so easy if it's not owned by the person requesting it.
But at least we know, like, a little bit what candidates might be likely to look at, right? Because going through it for millions and millions of records to do that is kind of a big ask, right?
Yeah, and I think part of, you know, I guess this is not really a solution either, is thinking about new submissions, right? Formatting the new submissions so that the database can see it came from OBI. You know, there's easier said than done, because humans are going to find 50 different ways to do it.
Yeah, and I noticed this really cool feature that is enabled on the search on NCBI, is that you can make an alert. So if there's a way to enable that, I think folks are using it, and yeah.
Yeah, you can set up a My NCBI search, and that would every week it would tell you whatever your search is. It will send you new things. But I was just looking at these vouchers. So OBI was just structured separately with a semicolon. You know, it would have. Because I looked. I mean, we have an entry for the herbarium, but there's only 2 sequences. That's linked to it. So that we're only aware of 2 sequences officially.
Yeah. And I have a feeling, just like if we stay focused on the herbariums that are, or the herbariums or any kind of collection that are eager to link, I think the linking would go a lot faster than trying to do everything at once, right?
Yeah, and I'm curious also to have some thoughts from everybody here. What's the way during the submission process that you could guide people to format these vouchers so that it's easier to find? I think that's also something that we're thinking about a lot. I'm not involved directly, but how do you ask without being totally onerous to a submitter to just give the right information in the right place so that others can find it? I don't know, that's a big challenge. I don't think there's easy solutions there.
It looks like Frank might have a comment to that. He has his hand raised.
Yeah, I was just going to say, if you borrow something from a herbarium these days, most herbaria have sent you a letter under which conditions you can use the material. I mean, after all, the herbaria supposedly own the material that they send out on loan. So part of these conditions could be explaining or asking that when you get DNA sequences that you upload to GenBank from the specimens, you could make specific conditions how they should be uploaded. So theoretically, that's not a big ask. I mean, you could essentially You can also negate anybody extracting DNA from a specimen if you feel that specimen is too valuable. For example, if it's a type specimen, very old, you don't want to have any damage done to the specimen, then it's pretty common that certain herbarias say, well, actually, you can have a look at it, but you're not supposed to do some destructive sampling. So that's a common situation. So including with this, you could ask or for specific ways how things should be uploaded, so.
Yeah, I am particularly interested in tracking the types, wherever I can find it. If I can find a way to make those links, that's a much, much smaller subset of links. That's probably an easier problem to solve than finding everything from it. If we can find ways to do the extra work on those, I think that's
some initial work that that I did on the RSA botanical garden. I think it's what's it again? The
California Botanic Garden.
Right. California Botanic Garden. There's a lot of low-hanging fruit there, and I was almost very almost tempted to just keep going, because it's such a bummer that like all that information is there, but it's just not picked up. There's just some links that that we can add to sort of make this useful information available.
Yeah, so if I can get a list of names and type specimen strings, text strings, and their various variants, collection numbers, whatever, field numbers, and various spelling variants, whatever, I could probably load that quite easily to our taxonomy database, and then the lookup processes can sort of propagate to the sequence records themselves.
Yeah. So if I provide the verbatim voucher text and then basically say this is what it should link to, is that sufficient?
I might have to get those records and then actually examine them and see what's on the record, because if the text string is not there, the link won't be possible. So now if the record was, for instance, was submitted without any voucher information, but you just tell me it came from a type, I'll need more evidence than just you telling me it's from type. I need to have, that's the difficult part. If there's no voucher information on the record, it's a very difficult problem to solve. If there's some sort of, submitters collection and the strings match, I'm not doing it as sophisticatedly as you. I mean, there's a lot of manual futzing, but I could probably improve that with some more thought. But,
I think most of the cookies I spent on developing the methods to scan through all these records. And so that's all there, and the corpus is there. So, doing this for another collection would be a little easier, I think. The only question is, who wants to do it and who's going to pay for it, right? Because I'm not on a payroll. I work for the joy.
We can start by people just sending me lists of accessions, and we can. I'm paid to do that kind of stuff, right? So it's part of my job description. So
cool. All right.
Accessions, and then the expected name that it's a type from, and the kind of type, whether it's a holotype or an epitype. That's essential to know. But it should be in all these herbarium databases. Maybe in GBIF. I mean, I've not really. Yeah. Anyway, that's the kind of stuff that could be done. Maybe it's not going to be easy, but it's possible. And then I think for us, then the focus is on submissions, see if we can improve the submission process. Maybe, as Frank suggested, maybe there's a recommendation one could make, and how a third party would submit something. Here's the format that we suggest you use. Of course, people won't do that all the time, but at least then there is some sort of a target that you could say, do it like this, and then it will link to us.
Ideally, I could see wanting to add the unique identifier, like the unique identifier, like the global unique identifier of a specimen record with its sequence. But of course, people don't always know where to find that. Frank, do you still have a comment?
I also thought one thing that might be paying for the cookies in a way is that if researchers can download data directly through a Symbiota portal. I mean, right now, what you mostly do if you're a researcher, you go to GenBank and you don't download FASTA files. And actually, for researchers, it's often inconvenient to actually understand exactly where that particular record comes from.
Mm-hmm.
I mean, for us, it would be really helpful from a use point of view to actually have access to, okay, where in which herbarium is that specimen deposited, and if I want to, I mean, sometimes people make mistakes, right? They like a lichen specimen might have 2-3 lichen thalli on the same piece of rock in the same packet, and the sequence came from the one on the left rather from the one on the right, but the one on the right is actually the one that the specimen was identified to. So it would improve the data for the researchers to be able to access that information. And so when you make okay, now you make the link with the GenBank record, but you can't download the sequences directly. So that would be an additional kind of cookie for the researchers, so to speak, that they could then harvest maybe directly from the Symbiota portal, you know. So and then the interest might be stronger for people using the Symbiota portals for that particular purpose.
Mm-hmm. Yeah, as far as I know, making a link to directly download that data is all the ingredients are in there now. So you have all the identifiers for the accession, right? So you can just make that happen, or just make that happen. I'd be curious to see how hard it would be, right?
So there are different downloadable options from GenBank than just FASTA files, which is not going to include a lot of the metadata. So I mean, if one wants to be a little bit more sophisticated than just grabbing the FASTA files, there's little scripts and not maybe go all hog like Jorrit. There's little scripts like source check. I'm not sure how easily it is to install it from outside, but it gives you all the accessions and all the metadata on the table. And you can look through that and start to manipulate that. It's not, you know, you need to do a little bit of putzing to get it to work, but it's possible.
So does it come down to trying things out, documenting the results and sharing them in discussions like this, and see how we can get better at linking?
Yep.
Yeah, probably. The question is, it might even require more outreach to a larger group, right? I mean, not just the people who visit these Symbiota support hub places, but people are members of different research communities, and then maybe I'm a member of what's called the lichen list server, so I could once these tools become available, share the tools through these forums.
Yeah, one of the challenges I have is that I see a lot of possibilities. I see a lot of ways to link data, and I also know that I also know that it takes time for tools to mature, right? And sometimes the first version just doesn't work at all, and then the second version is a step backwards even from that, right? So it takes a little time to mature them. So do you have any thoughts on how to make sure that the tools are mature enough to be shared outside of the friends and family, right? If you know what I mean.
I think we're getting close to the hour.
Okay.
Thank you. So I'm going to just finish up our slides really quickly, and I can stick around at the end for anybody else who wants to keep talking about it. The slides won't take very long, but just wanted to point out we're almost at 1 Pacific. So hold that thought, but we can continue talking after I finish the slides. All right, so slideshow.
Also, Libby has a question in the chat, Jorrit, you can look at and think about.
Right. So quickly, I will go through other methods for linking genetic data in Symbiota or in GenBank to Symbiota. And then we'll talk about extended specimen, and then we'll just wrap up really fast. But we've hit on a lot of this already at this point. So, you know, we've talked, Jorrit's discussion is a lot about like mass and like bulk attaching things, but there is the brute force method of doing this one by one. So if you go into GenBank and you like see, you know, a record in there that you want linked in Symbiota, you can do that very easily by just going to the occurrence editor, going to linked resources and clicking on the linked resources tab and then scrolling down, and there is a genetic resources box where you can edit this. So I'll just demo that really fast. So you're gonna go to linked resources, go down here, and then you can add multiple records if you want. And I'll point out that this is not just for GenBank, there are other databases out there. So what's showing up here is just literally what you're typing in this name box. So if you know, have this, get this information from another database, you can enter that here and it'll show up on the record publicly too. And then somebody brought up in the chat the same question about how do I do this for a lot of things? So there's no user interface right now for batch linking these records. You would have to send us a spreadsheet of this information. So for example, if you had a spreadsheet with a column for name, a column for identifier, locus, URL, and notes, you could just send us that spreadsheet. We can batch attach it for you on the back end. Just the really important thing to include is the catalog number so that we know which specimen to associate everything with. And then something not everybody may be familiar with is we just want to point out that if you haven't tried this before, you can find records in Symbiota portals using a pretty simple limit on your search. And so, for example, we can do this in CCH2. So you would just go search like normal. So we're going to search for all the collections in this portal. You would limit to specimens with genetic data, pull up whichever display you prefer. And in CCH2, it looks like there are 316 records that have genetic data attached already. And congrats to Genevieve, who asked this question in the chat. Looks like her collection has a lot of those linkages already, so that's awesome. And then just bringing this back to the extended specimen. So for anybody who's not familiar, we've talked about this in previous Symbiota support groups before, but the extended specimen is really the concept that, you know, your single specimen extends beyond the singular physical object, and genetic data is part of that. So if you look at the figure on the right, that is pretty close extension to the primary extension of the specimen. So that's, you know, we try to support the extended specimen in Symbiota. And this is one example of how we do that. And so if this is a topic that you're very interested in, I recommend going and checking out the previous recording of the support group. We did on this in April. Similarly, the workshop recording that Jorrit pointed out in his presentation, the link for that is here. And I think I put it in the chat already. And then, of course, our website has a list of all the ways that Symbiota supports the extended specimen. And so where it all started, I think I linked to the exact moment in the chat where this conversation happens. And if this is a conversation that does interest you, I do recommend going and listening in on that. It brings up some really good points, very related to what we've been talking about here today. And then lastly, we've also talked about this a little bit, but to help Jorrit with his cookie-nomics, you know, this development is not free. It takes a lot of time and energy, but a lot of the work has been done. So if anyone out there is really interested in doing this for their collection and you're, you know, looking to write a future digitization grant or you're working on it already, this would be a pretty easy thing to incorporate into a future project. So not only would your collection benefit, but so would the entire Symbiota user community and beyond. And with that, we can wrap up. So if anybody wants to stick around for more discussion, I'm happy to stay on the call, but I realize it's pretty much at the top of the hour, so you have to head out. We'll be recording this. You could always come back and watch.
I just wanted to acknowledge the conversation that's going on in the chat. Libby was asking what kind of options are there for automation of this process in the future? And I think the answer is what you just referenced, Lindsay, is that we need tools that can do this in an automated fashion, and we don't have those. So what we need is the funding and the attention to this in order to make it happen in some future grant or some future, you know, funding option. And Jorrit makes a good point that you still do need some sort of review. Like, I don't know if this is going to ever be fully automated, and unless GenBank was fully Darwin core and they just lined up really nicely. But, those are two very different research communities. And so I don't think they're ever going to fully mesh with one another. But I think that there are probably ways that this could be more automated. Like, you could look at a list of like all these potential duplicate or potential link ups, and there would be a way to just be like, check, 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 bring those all in. But those tools don't exist yet. So it would be nice to have those.
What you described, Kitty, sounds a little bit like a bionomia for this other kind of linkage.
Precisely. Yeah, that's exactly what would be needed.
Yeah, so I would say that the tools are there, and I agree that there's no good user interface for it yet. For folks that would like to, are more familiar with bionomia, like user experiences, which is like super nice, right?
Yeah, like the software tools are there, but when I think of tool, I think of like an herbarium curator could go and do this just like in an afternoon. They're not gonna download us. Like, I'm a database analyst and I don't go in and do random scripts and pull them and then do comparisons on a regular basis. So a UI that allows any person to do that.
Yeah, that'd be cool.
Broader impacts, next NSF grant.
Also, I think I kind of tend to disagree that those are necessarily two research communities. Yes, they are at the moment, but I think there's a lot of talks that I've seen already where people are talking about getting DNA out of really old specimens. And so in theory, and I think we all hope that this will eventually happen, is that there will be technologies and DNA stuff is advancing really fast, whereby you could theoretically sequence a whole herbarium or something, right? I mean, that's beyond what is possible right now, but it's not unfathomable that this will happen at some point, so.
Yeah, absolutely.
Alan, Greg, did you have questions?
I just had it's somewhat related, somewhat off topic, but I love that in the search command, you can sort of filter out what has genetic linkages within a collection. Is there plans to do something similar for whether there's tissue associated? It's sort of the first step to getting the sequence linked is letting researchers know what specimens have available tissues. I would love to be able to filter my search in Symbiota portals by what has available tissue. So this would be a material sample type filter.
I think that does exist. I'm not sure, actually, now that I think about it. It's probably being developed for the Neon portal because they have a lot of material samples. So I'm sure that's on the development task list, if it's not already done.
Awesome. I have one other question, or something to throw out is a lot of the publications coming out now are next gen sequence, so they're not even going into GenBank. Is anybody exploring how to link sort of genome wide sequence, because they don't seem to be quite as structured as GenBank? Maybe because it's newer, there've not been a consensus on how that information is shared online.
Where are they deposited? Are they deposited anywhere?
They're usually deposited as zip files in places like Dryad or Open Science Framework. Yeah. So
yeah, I would encourage you to create examples, document them, and share your desires, and then who knows, the universe might give you some
provide cookies.
Well, yeah, but I think, like, as I'm trying to get better at describing what I do, I'm sort of realizing how little I know about the challenges that folks in collections have today. So if there's more people saying, hey, wouldn't it be great if, and then give some very specific examples,
Yeah.
I think a lot of cool things can happen. Yeah.
Agreed. Cool.
Is that it for today? Anybody else going once? This has spurred a lot of conversation. Well, I guess not. So I just want to say thank you to Jorrit for joining us and giving us the presentation. That was really awesome. Thanks for being our very first Symbiota support group guest presenter. The very first to do that. We do, you know, if anybody else wants to, we're taking suggestions for future topics. But Jorrit, that was great. Thank you so much and for all the work you put into this. We
Yeah,
really
thanks for having me.
Thanks, everyone.